We next have Dr. Larry Reiter, who is going to be looking at delayed neural development in PWS neurons. So Dr. Reiter's group, for those of you who have been following FPWR for a while, his group has been studying neurons that come from PWS baby teeth. So many of you have sent your child's baby teeth to the Reiter lab, and they use that to grow out neurons. And it gives them a great source of neurons from lots of different patients. So they're able to look at the molecular characteristics. And they look at PWS neurons, but they also look at, typical neurons and then other neurodevelopmental disorders. So they have a really nice bank of neurons to look at. So what they found, and what we've heard a little bit from other researchers, is that the PWS neurons seem to have a different timeline of maturing than typical neurons. And this may be indicative of an underlying issue in PWS that is related to some of the social and cognitive and developmental issues that we see. So with this study, they're really going to focus in on that, looking at a larger sample set across more patients. And the team is also going to be taking genes and pharmacological agents and seeing if that can restore a more normal maturation process in these PWS neurons. So we're excited about this study. This is a researcher who's been quite productive and has really used those baby teeth that you all have sent to really begin to understand the very basic biology of how a PWS neuron is different than a typical neuron. So this is a unique resource that's being used, and it's a phenotype, or it's a characteristic. This delayed maturation is a characteristic that can potentially be used in drug screening and to assess the efficacy of a treatment in a dish. So a relatively easy way to look at new compounds and see if they might correct this difference in PWS neurons. So we're looking to this work to define targets for new therapies aimed at releasing this developmental block and restoring the PWS neurons to a more typical maturation profile.